our first speaker of the afternoon is going to be Xingyuan Chen, and she's going to speak about omics-informed reactive transport modeling in watershed systems. So I think we're maybe a minute early, but I don't know, 12:29. I think you can go ahead and start, Xingyuan.
Or like I can wait for another minute to take a breath and checking if you can see my mouse if I move it around.
Yes, I can see your mouse if you move it around.
Okay, that's nice.
Everything looks good, and my clock says 12:30, so let's go.
Okay, thank you, Tim. Good afternoon, everyone. And my name is Xingyuan Chen. I am a research scientist here at PNNL. My daily work is simulating the hydrologic and biogeochemical processes across watershed systems. And I also do a lot of model data integration that we apply like statistical methods, including machine learning to improve our predictive models. So the presentation I will give you some examples of what we do with this omics data to simulate and quantify the cumulative effects of like denitrification or respiration in the river. And it involves like work from a big group of colleagues we work over here. So I would like to start acknowledging the contributions from all of them, including Tim, who has been the organizer for your great summer school. And also like a lot of results come from the project he is leading with a lot of scientists over here. So it was really nice to hear the flash talks from all of you this morning, and you might see how your work might plug in to this kind of a like bigger scale hydrobiogeochemical studies. Let's see. I might have clicked it two more times, but so when we talk about watershed function, we're talking about the complex hydrologic and biogeochemical processes happening within a watershed. And it's impacted from processes happening at molecule scale all the way to a watershed or basin scales. For our study, we especially focus on a narrow band around the river, which we call river corridor. Within that system, the characteristics is being the hydrologic exchange flows. We define that as the bidirectional exchange between the river water body and the surrounding environment around it. And then what this exchange flow does is it changes the thermal regimes of this interface and brings different water bodies together that creates mixing between different water chemistry and also stimulate the microbial functions within that. So this kind of a hydrologic exchange plays a very important role in controlling the transformations of key nutrients and contaminants in this river corridor system we're going to focus on for the rest of this talk. And there are like significant knowledge gaps in understanding this complex system. So you can see this upright over here is a real system at the Columbia River Basin we are in here. And this is how we conceptualize the system based on our understanding. It's like who are in the system, what they do, how they interact with each other, and how do the external drivers like extreme precipitation or fire impacted how everyone in the system is functioning. So a big challenge is for us to understand and also quantify processes governing the cumulative effects of the biogeochemical processes controlled by like HEFS, the organic chemistry, microbial activity, and disturbances. To to deal with that grand challenge, we need a key over here is to translate the understanding at smaller scales all the way across different scales to the basin, to the watershed or the basin. We're interested in quantifying the cumulative effects of biogeochemical processes. And you have learned a lot of like theory and the tools that can help us to understand what's happening at the molecular and a column scale to figure out what reactions happen, how fast they happen, and then with that we can scale up to quantify what we might predict at the basin scale. With that kind of a knowledge in place, we can answer questions like what watershed characteristics control the spatial and temporal variations in river corridor biogeochemistry, and with the disturbances like wildfires, how do the river corridors mediate the watershed responses in both quantity and quality to wildfires and precipitation events. To deal with this like very complex problems, we usually start with the hydrology first before we couple hydrology with biogeochemistry, so we can understand what's the driver of the biogeochemical functions of our system. To give you an example of a watershed scale hydrologic simulations we have performed, this is a, actually we are about over here. And this is like a regular size watershed we carved out from the bigger aerial image of the Columbia River Basin. And our model domain is 60 kilometers wide and long, 200 meters deep. And we use the P-flow trend to simulate how do this like heterogeneous hydrogeology combined with river geomorphology with the fluctuations of river stages in this different places along the river, which is like regulated by a dam upstream from our domain. And how do they all like work together to control, to determine how much exchanges is occurring between the river and the groundwater aquifer system, and how do they vary in time as well. So we use the P-flow trend to do that. And more details of this model can be found in a recent paper Ping Shui published. And for this model, we discretize this whole domain into grids of size of 100 meter by 100 meter horizontally and 2 meter vertically. In total, we had about 36 million cells. And we are applying uniform property for each material, like each color means one same hydraulic conductivity. And we applied like no boundary conditions across the lateral and at the bottom. And the river stage is taken, is like varying as shown in this figure. We took that from another river routing model that gave us a very accurate prediction of the spatial temporal dynamics of river stages along this region. And we are simulating the whole system in like 6-hour time step in maximum. And then this is like what we see from our model. So we, on this animation, we're only showing the exchange fluxes between the river and the groundwater. So it's a flux across the riverbed. The red colors mean the water is going from the aquifer to the river, so river is gaining, and negative means the opposite. It is like very obvious from this animation that there are certain regions you see like more exchanges than the other regions, and also this kind of heterogeneity, they change when the river stage goes up and down in time. Then we need to know, it's like, what does this kind of exchange mean to where the mixing happens? So what we did was to apply numerical tracers in the river, and we track where they go in time and visualize in this animation, similar to the exchange flux animation. The redder color means more river water in the groundwater system or in the river, and the blue one is more dominated by the groundwater. So you can see over time, you can see those hot spots of exchanges that's indicated by like the presence of more river water in that place. And you can also see how this zones of influence change over time, driven by the river stage, regulated by the dam upstream. And you can also see the heterogeneity is playing a very significant role in controlling where the river prefers to go. And for example, like across the big meander over here, there's also a preferential flow path of exchange that is promoted by a paleo channel we have identified from like geological records from all those wells that have been drilled in this system. So if we zoom into like this region for more details, you can also see how this exchanges and mixing are changing the temperature field of at the riverbed. So the warmer color means higher temperature, and red ones are colder ones. You might notice that groundwater temperature seems to be pretty stable because that's what we observed from our monitoring network. And that's also how we conceptualize our model in boundary condition to make groundwater groundwater temperature pretty stable over time. And then you can also see how the fluctuations in river temperature is actually also captured by our model that is strongly related to the exchanges that we have captured in the numerical model as well. And the temperature simulation is very important for biogeochemical reactions because they change the rates of all those reactions. And the next question is like, how do we then simulate the biogeochemical processes in the system? And the first thing we need is like, we need to know what reactions and how fast they occur. So we need to put in a reaction network with the kinetics. And we have also learned to capture such like reactions. We need to do very fine scale hydrobiogeochemical and thermal simulation. So you can imagine we cannot do that with a 100 meter by 100 meter and 2 meter resolution as we have done for the hydrologic exchange flow because not everywhere is reactive. So what we did was to first see how the biogeochemical hotspots and hot moments look like within a refined domain with very high resolution. So we pick like almost a grid size from our reach scale model. I mean, this domain we study in detail is about the size of a grid cell in our Watershed scale model we just presented. And then we took a 2D cross section which incorporated all this like fine detail heterogeneity in our geological layers, which are like mainly composed of 3 types. One is the, we call like, alluvial layer. That is the more recent deposit at our riverbed, which is the most reactive layer of our system as well. And then we also, like beneath that, we have a very high permeable Hanford formation, which are having like a hydraulic conductivity in the range of like thousands of meters per day. and these two layers are underlain by a ringgold formation, which is used as like our confining layer. And we, like for this model, our resolution in the riverbed is about like 0.1 meter vertically and horizontally. And at the same time, we also drive the boundary condition, like the hydrological boundary condition on both ends using fluctuating river, river stages and groundwater level we obtained from our observation. And now what we have seen from that model is this. Sorry, it's a very busy animation, but what we're showing over here is on the left, we're showing the concentration of all the species we incorporated into our model. On the right, we're showing like the reaction rate. That means it's either production or consumption of that species at that time and over space. So this like jumping line indicates where the river table was at a given time point. So although it's a very busy, but sometimes we like to see this because it gives us not only the spatial heterogeneity, but also the dynamics of the concentration and also what's reaction, like the reaction hotspots very intuitively. And we can see how different species are responding to the river fluctuation differently as well. And so this level of high resolution simulation is really critical for capturing the heterogeneity of the reactions within our system. And as we know, we will not be able to do this level of a simulation across a watershed, even if we only focus on the river corridor. So what we do, do you see a blue screen right now?
Not yet. I'm seeing your, oh, now I see it.
Okay. I think something happened on my end.
Yeah, it looks like it crashed.
Yeah, let me restart sharing. Maybe my movie was like too busy and the computer doesn't like it. Let's see. Sorry about it. How is it?
Pretty good. It's cutting off there. It looks good. Okay. Yep. Looks good now.
Oh, okay. So now we're talking about how can we capture all those like hot spots and hot moments of biogeochemical processes at the river corridor scale. So we definitely need to do a lot of simplification so that we can simulate them at that level. So what we did is we conceptualized a grid cell or like a river corridor unit in here. And for that unit, we will use a reduced order model to characterize the hydrologic exchanges between the river and the channel. And we specify the same set of reactions across the entire unit, and we apply the reaction rates that's from like lab experiments and field experiments. And we found like we have to scale those reaction rates so that we can get close to what's really happening in the field. And the reason being, like, if you remember the movie, it's like the reaction rates are very different at one place to another, especially like beyond the riverbed, the reaction rates are a lot slower. So that's the reason we have to scale the reaction parameters to like lower rates when we are dealing with like bigger units at watershed scale biogeochemical modeling. And we also found from one of our applications at a similar watershed scale, we simulated hydrology and we showed the animations that what is processed at a given unit depends on how much nutrient inputs that is present adjacent to that unit, and what kind of exchanges are happening between the river water and the groundwater. So we built this like river corridor model that can allow us to specify the processes within a unit and link all those units together across the river, like across this watershed, to quantify what's happening at each place. And then the question is, how does all the theory and tools you have learned from this summer school apply to that scale as well? So I am not going to repeat this slide, because I believe you have heard enough from how you go from the omics or FTICR data to generate a reaction network we can incorporate into the reactive transport model. And you have like both Han and Kelly, they are heavily involved in this process. And all these like pipelines are built in collaboration between like multiple projects funded by the Subsurface Biogeochemical Research Program, one of them being the Ideas Watersheds. If you have heard like the brief introduction I did before lunch, Ideas Watersheds are aimed to build like simulation tools as well as workflow that can facilitate the assimilation of information from a useful data source and a theory to inform our hydro and biogeochemical models. Then what that new theory and tool has made a difference is, like, remember I showed you like three or four reactions for that high resolution model we performed simulations. With this new omics or FTICR information combined, it can generate thousands of reactions we can include in our reactive transport model. Of course, we're not able to like incorporate all of them, and Han helped us to like down select a reduced set of reactions we can use, which is like 10, shown over here as an example. So you can see the reactions are a lot more, complicated. It's not only in terms of the number of reactions, but the species are, there's like more details in what kind of an organic carbon species is in each like reaction. So the our hypothesis would be like capturing all those nuances in this reaction network along with the reaction kinetics. We will be able to better capture the hot spots and hot moments of reactions within our river corridor systems. And we can use the observations to validate our assumption and also test our hypothesis. Then this gives you a high-level overview of how this is. Sorry, let me mute my Skype. So this highlights our like entire strategy of incorporating the small-scale information from those omics and the other observations through the pipeline to generate the reaction networks and the kinetics to inform our river corridor model. And that river corridor model can be integrated into our watershed and basin scale river corridor models to help us reveal the spatial and temporal patterns of reactions. And then we actually also use that information to guide where and when to collect our samples that can make the most impact to improve our models. This is a cycle we, if you talk to like our colleagues here, we often mention this as like the MODEX. It's model experiment iteration MODEX cycle, or it's model data integration, underpinning how we improve our predictive models. In more implementation details, our goal is to quantify the carbon and nitrogen cycling within the river corridor, and we will be the spatial patterns may look like the two we provided as an example over here. It's the output we get from our river corridor model, and this model requires 3 major inputs. One is solutes in the river over here, and then we need to know how much exchanges are happening between the river and the groundwater. And we also need to know what reactions and how fast they occur at each place. It's a lot of work to link all of them together, especially if we want a lot of fine details plus fine mechanisms. That's why, like in reality, when we're doing all this work, we take simplifications, or we start from a simple place. For example, we start with steady state inputs to our system and steady state exchanges. We can use a reduced set of reactions that do not change over time and over space, like the one we used in our like high resolution 2D simulations. But then the improvements we can make is like we can we can bring in heterogeneity in the system, and we can also bring in like dynamics in inputs to our river corridor through like integrated watershed modeling, which we can talk later. And to the most interest to this group is we can enhance what's happening within the river corridor and how that is impacted by the other things we can measure, like dissolved organic matter, pyrogenic organic matter, and water chemistry and microbial functions to have a say more accurate representation of our reaction mechanisms. And you might figure out like this line, it's from this microbial function and all those information to generate reaction mechanism and put it into a model, and then use to quantify our like spatial patterns of this cumulative processing of carbon and nitrogen is the main scaling line we have presented earlier. So this gives us one-time snapshot. If we do this over time, what we'll be able to see would be like if we sit in one place, how do the concentration of nitrate or other species change over time? And this is like what we can observe in field, like USGS is doing a lot of such monitoring, and then we can use that to evaluate how good our model is capturing such dynamics. There's also the other thing we can do with the spatial predictions we have used our model to generate. One interesting question to ask is like, what is controlling such spatial pattern? And we can imagine it's like the riverbed physical properties and land use, land cover change of the basin, and also the climate variables. It's like how much rainfall and how warm or cold it is in each region. They might all come into play in controlling how much total nitrate is being removed in our system. So one way to answer this kind of question is to take our model inputs and outputs and run through a like classification method, like machine learning based random forest model, to see which variables are contributing or are explaining the most variability of like the spatial patterns of our model outputs. And we did like some analysis like that, and this is like the example of how random forest is helping us to classify our like this would be like the outputs the model can generate, and those are the variables we suspect are playing a role. And then from the results from that, we can see first how the statistical or like machine learning model is capturing all these predictions we generated from our river corridor model. In this case, it's pretty good because the R square is like 0.9. And you can also see most of the points are along the one-to-one line. And we can also look into details of like which variable is explaining most of the variance. So from this case, like across the Columbia River Basin, we found the riverbed physical heterogeneity and the land use control is the most dominant factors in determining the spatial pattern in total nitrate that is removed in our system. Then the next thing, so that is work we have done. And then the research we are planning to do in the future is really to use like distributed sampling of all this DOM, PyOM, and water chemistry and microbial functions through wonders, like through the network like wonders. We collect through the system, and how can we use that information to inform our basin scale river corridor models. If you're still with me, you like what this can impact is really the reaction mechanisms we can map across the basin scale. And then what we need to do is connect from those kind of distributed sampling to a distributed mapping of the reaction network and the kinetics across the entire Columbia River Basin. So this is how we can utilize all those pipelines we have been building through like multiple projects by collaborating with our fabulous community. So what we will do is with all those samples, we will extract the patterns in our data, and then we run those extracted patterns through our pipeline that will give us the distributed the point samples of what the reactions and rates are looking like. And then we build, like, say, some machine learning models that can help us to map what we can measure or what we can get from elsewhere to the reactions and rates we need in our model. If we build this mapping, then we will be able to use all the information we can get. We can take full advantage of distributed sampling of this like wonders network to make our basin scale predictive models more accurate. So, and also like with this, we also talked about the other way to improve the river corridor biogeochemical model is to know better about what comes into our river and what is controlling the spatial and temporal dynamics of such inputs. If you have heard, I talk for ideas, and this is, going to be a repeat of some information from that. So our Watershed model can help us to accomplish that goal. But the key over this, the key of using Watershed model to do this is to couple all those processes that are contributing to what ends up in the river in a mechanistic way, and also can be computationally effective. So we will be working very closely with the Ideas Watershed project team to leverage the ecosystems the community has built for different purposes. And we can build like interoperable interfaces to allow us link different processes from different codes together. And by doing that, we will be able to build a single code that can have all the processes we're interested in for this system. But this doesn't mean everyone has to use this code. You can have your own choice of you can have your own recipe that is best suited for your problem. For example, we can have P-Flowtran to simulate the biogeochemistry, and we use ATS. It's another like code developed by our community to simulate the integrated hydrology for both surface and subsurface. And you can use alchemia to link them together. And then you can choose WRF hydro or ERM for your land surface or like plants related processes. And it's very important to perform rigorous like comparisons across different model configurations and compare that with data to help us decide what is the best model we can possibly build for a system, for a variable we're interested in predicting. And with this system in place in the end, it's like we want to like currently our river corridor is often treated as a separate component from the watershed, and we do pass like mass and solutes across those across river corridor and the rest of the watersheds. But in the future, we would really like to build an integrated model that we refine our mesh in the river corridor, which we consider as the biogeochemical hotspots, and it's in one system of like with the rest of the watershed model. So this is really what we're trying to accomplish. With that kind of integrated simulation capability, what we will be able to do is to capture how do the transport pathways vary under different conditions. It can be under a baseline condition, but can also be after a fire. And what this transport pathway does is like it will bring different materials from different places to the river. And when they change, you can imagine the composition of the chemistry in the river will change, and also like the timing of like the concentration peaks or certain signatures will change because of the change in what transport pathway as well. And we would like to know how the changes in microbial function and also the organic carbon speciations will be coupled with this kind of a hydrologic changes to change the river corridor biogeochemistry as well. So with this, we can test hypotheses like what's controlling the short-term versus long-term effects of extreme events like wildfire combined with extreme precipitation. The simulation capability opens the door for us to do that. And if we do our simulations to like say, systems that are disturbed versus not disturbed, we can do paired watershed analysis to analyze how do the fire history and precipitation scenarios interact to alter the hydrobiogeochemical regimes in the river, and how does that change the river corridor biogeochemistry in the end. Like I have mentioned, in order to do a good job over there, we have to integrate our model, or we have to use like all the information we can find from a wide variety of measurements like the USGS monitoring stations, Ameriflex towers, remote sensing, to parameterize our model and also to evaluate and improve our model. So we plan to use a lot of machine learning methods to help us integrate the information in our model and in our data. Eventually, we build a great model for understanding the like river corridor biogeochemistry. So with that, I would like to say this is a daunting task to do it well. So that's why the community involvement and like to work with everyone who is interested in studying such systems and such science problems are very important. By doing it together, we will accomplish way more than what we can accomplish individually. And that, I believe, is also the purpose of doing this, to have this summer school, so that we can build a great community that are centered around the common goal. And then we share our capabilities, we share our data. In the end, we can understand much better of the spatial heterogeneity and the dynamics of our system, and how they scale from a single point to a watershed and to a basin, and also to the entire United States or the globe. Wherever the wonders are taking us to sample, we want to model with them, so we get the best out of a distributed sampling network like wonders. And with that, I would like to thank you all for your attention. And it's really nice to share like our research with you as well. So I'll take questions later. Thanks.
Thank you, Xingyuan. That was great. I really appreciate the way you concluded there, because I think you're completely right. This summer school this week, I think one of the biggest outcomes is really coming together as a community, demonstrating to the participants, the attendees, and the students, what a community can do together. When we sort of set aside our institutional boundaries, or our all of the things that tend to isolate us. And so this has been really, I think it's been really cool, and your talk was a great illustration of that, so thank you. There have not been a huge amount of questions so far, at least nothing in the Zoom chat. There's some questions over in, let's see, there's some questions over in the applications area of the Discord chat. And one in particular is asking about publications related to the models that you presented today. So I know you have some papers you can share.
Mm-hmm.
So are you, if you're in the Discord, if you could head over there when you have a chance and check out those questions and share some papers with the people.